Hey, hello, welcome to my first video. My name is Rodrigo Moreno, and I am a PhD at Rice University. I am currently doing my postdoc, and I want to start a new channel in YouTube called Moonlight Molecular Biology. This channel will be all about uh, talking about the molecular biology stuff. So that means I am going to look for papers that seems to be very interesting, new data. I'm going to try to find any interesting news, and I am also going to try to look for like some interesting techniques or stuff like that. So welcome, and yeah, as you can see, I am showing you my screen. It's a Feedly web page, and in this web page, what I am doing is just retrieving the new data, well, the new papers or new news that are related with molecular biology. So, for example, at my left, you can see here, actually, I will try to increase my resolution so you can see, and I will move me to the right side. But overall, you can see that I am following a lot of very interesting molecular biology web pages. And overall, what I want to do is to show you any interesting thing that we can see overall throughout the week. Usually, these are the news that I have been looking. Like for example, today it's there. I have different feeds from, for example, bioRxiv. Then I have some others from. Let's see. This is all bioRxiv. So it seems that we're looking too much bioRxiv here, but if you can see here, when I load all my molecular biology, we can start to see that there's not only bioRxiv, but it's also Science Magazine, molecular biology news, and so on. So we can try to find like an interesting paper that we may see. Usually I like to find cool figures. Like for example, this seems to be very interesting, and this has been just recently published like 9 hours ago. So this is the one that I actually will love to see because it's a cryo-EM structure of human telomerase ribonucleoprotein. So we are going to access to this paper. It's a Nature paper. So I will accept the cookies and we can try to see how it goes in here. So yeah, let's see how it goes. Usually what I like to see is the first figure. This is usually like the most beautiful figure. And as you can see here, we are, well, the overall idea of all this live streaming is to find like the papers, try to see if I am able to understand them and I am able to show you the interesting stuff. But yeah, so let's see. Sorry, it was my phone. So yes, let's continue with this. Let me move my camera a little bit to the left so you can see well how I look when I am in the camera. So yes, it seems that you can see me now. Yes, that's how it should be. Okay, so yes, like for example, as I was telling you, what I like to see is the figures of the paper overall. Maybe obviously to see what's about the abstract. So it's about the telomerase ribonucleoprotein. Ribonucleoprotein, as you can remember, is a complex based on RNA and proteins. And this seems to be, well, as if you cannot remember, the telomerase is a protein that adds as far as I remember, it adds the telomeric repeats in the eukaryotic ends of the chromosome. So let's see, it says that it has two functional lobes, so that's HTR and a hinge, called hinge and HCA, means HACA or HACA. This is a ribonucleoprotein, and this seems to be responsible for telomerase biogenesis. So let's see, this is critical for the biogenesis of the spliceosome and ribosome. That seems to be very interesting. Okay, so it seems that this telomerase seems to be having a general role in pseudorylation and spliceosomal and ribosomal RNAs. So that's very interesting. Let's see, it says understanding of eukaryotic HACA ribonucleoproteins of human telomerase. So it seems that they report a 2.7 cryo-electron microscope. That means it's a very good resolution based on cryo-EM microscopy. And then it says that it improves from a previous 3.3 to 8.2 Armstrong structure. So that means we are going to see way more detail about this telomerase. And what else? It seems that we are going to see overall what about the structure. So obviously we are not going to read the complete paper. Maybe we can see something overall, but what I usually try to see is the figures. Sometimes the figures talk by themselves. So you can see here as actually is very interesting because it notes by color how is the structure of the human telomerase. Ahorita ya no hay, pero juega pato. So yes, that's my daughter asking for Pockies. So that means some chocolates that she loves. So let's see, this is the telomerase ribonucleoprotein. And it seems that it's divided in two sections, the catalytic core and the one that we were talking about, the HCA ribonucleoprotein region. And it's very easy just to follow the colors. This line seems to be the RNA. So this is an RNA that folds by itself and creates a secondary structure that folds. And it creates a pseudoknot, then goes to the HACA ribonucleoprotein region. And as you can see, it bends and then goes back to the catalytic core, goes back into a 5' hairpin, and then there's the H box, some other secondary structure where it seems that there's binding of different proteins, like for example TCAB1 and HACA heterotetramers. So this is very cool, very interesting, and we can try to follow the colors. As you can see here, in here we can see the blue parts that are means the HTR regions probably are going to be the TCAB1. Okay, so these are the proteins. Then let's try to find what else is in here, HTR. So yes, I am still cannot see at the first glance what's going on, but if we see in here, this is like more cartoonish style of this telomerase. We can try to see that overall this is very complicated to be true. But then we can start to see in this part, in panel number E, the overall difference between RNA and proteins, as you can see here. So RNA is the blue part. As you can see, it folds by itself. It creates loops, and then it turns back, and so. But the important part is that it interacts with several proteins like this one, this green GAR1, 3' GAR1, 5' GAR1, NOP10, TCAB1. So it's interesting. There's actually a lot of proteins in here in this human telomerase. And then let's go back to the paper just to see. Obviously, this is the figure number 1. Then we are going to see more results. So this is super cool. We can see probably here is the protein interactions and the structure of the specific proteins that bind to the telomerase. It seems that it's the human telomerase. And then if we continue, we can see the increase of interaction. So yes, as you can see, each panel is very interesting. because there's a lot of interactions in here, a lot of RNA and protein interactions, and there's a lot of domains. There's also, like, as you can see here in these panels, there's how there are different protein characteristics based on the hydrophobic and hydrophilic electrostatic potential, so that's very interesting. And in here you can see even at a more molecular level how each of the atoms or each of the amino acids interacts with, like, I think probably this is like the RNA structure. And then in here you can see here that there are more like in detail interactions between the RNA and the proteins, but overall, as you can see, it's like a lot of detail based on the different parts of the human telomerase. And then here, way more interactions. So this is probably the RNA that it folds by itself. This represents that it probably go back to the protein, and there's here a lot of like interactions between the different proteins that we usually see. And this is very, very nice figure because you can see here the different nucleotides based on obviously RNA. And you can see here like it seems to be this is the cytosine, the adenine, cytosine, and this is the uracil. And as you remember, the RNA is made of cytosine, adenosine, guanine, and uracil. And uracil is instead of thymidine. So you can see here very, very nicely how this part of this sequence of the RNA of the human telomerase actually binds into these protein pockets of probably the ACA box. So yes, that's very, very cool how this interaction works. And this is just another, yeah, as you can see, I am just trying to see what's going on with this paper. It's super fast, but we are seeing overall that there's very, very nice figures, and that's very lucky for me because this is the video number 1 of the Moonlight channel, molecular biology channel. So I'm going to try to explain way more papers, way more figures, and I am going to try to find, like, even in the future, maybe to have some interaction with some of the authors of the papers and so on. But I hope that at least with this first video, that let's see how long we have been here. 12 minutes. So 12 minutes is nice. It's a nice length for the first video that we will have in this channel. And I just want to remind you that I would love you to start follow my channel so we can start to add more content, to see more interesting papers and so on. But yeah, as you can see, this is what we are going to do, like from today to the future. We are going to try to find new papers, try to see what's going on with these ones. We are going to try to go a little bit more in detail. We can also interview the scientists that are behind these papers. But yes, to be able to do that, I will need you to start liking the video, to start following the channel, so we can try to understand more of these papers that seems to be super interesting based on based on the newsfeed that I am having, that it's all about related molecular biology. So, well, this is the first video. Don't forget to like and follow. And with that, I want to, I will say goodbye for today. But that was the first very interesting video that is based on the structure of let's go to the top. It's called the structure of the human telomerase ribonucleoprotein. So yes, let's see just the authors, Gani and et al. That means the other authors. It's a Nature Communications paper 2024, and that would be all for today, but let's try to make another video in some other day, okay? So see you and have a good night. Bye-bye.